about penguin toll-like receptors.
Thank you very much. Hi, everyone, and thank you for having me. I'm Stephen Fiddaman, and I've just started a postdoc in Oxford working with Adrian Smith, who's been speaking earlier today. So today I'm going to talk to you about a project I've been working on for the last 18 months or so, which looks at a cryptic pseudogene in penguin TLR15. So as a very brief recap, because I know TLRs are covered several times in this session, penguins, like most other birds, have 10 toll-like receptors, and of these TLRs, 3, 4, 5, and 7 are one-to-one orthologs with mammalian TLRs. And birds have undergone 2 tandem duplications of TLR1s and 2s, so they have 2 TLR1s and 2 TLR2s. In addition, birds have TLR21, which is a CPG recognizing TLR that is functionally analogous to mammalian TLR9, but the 2 TLRs don't share a common evolutionary origin. Birds and reptiles also have another TLR, TLR15. TLR15 is relatively understudied compared to other avian TLRs, but it's now known, as Adrian talked about before, it's now known to respond to a yeast-derived agonist, or somewhat unusually for a TLR, cleavage by microbial proteases, which I think, Kathy, answers your question. TLR15 caught my eye as being unusual in penguins for reasons that will become clear in the next few slides. So this is the gene structure for TLR15, which is a single exon in penguins. And TLR15 has a very typical TLR architecture. It has an N-terminal extracellular domain, which contains leucine-rich repeats, which are responsible for binding the agonist. It has a single transmembrane domain, and it has a tear domain, which is responsible for downstream signaling. We published the genomes for all extant penguin species last year as part of a large consortium. So I decided to carry on from our previous work in the Gen 2 penguins that Adrian mentioned earlier by looking at all penguin TLRs across all species. However, when I was doing this, I noticed that TLR15 was a pseudogene in 7 out of the 8 species of penguin in the Eudyptes genus, which are the crested penguins like macaronis and rockhoppers. But the strange thing was that the pseudogene causing mutations were often different in different species, and they were distributed all across the coding sequence of the gene. So this implies that TLR15 is not only a pseudogene in the Eudyptes penguins, but that it has been independently pseudogenized several times. And this would be highly unusual in a single genus. So I investigated that a bit further. So to rule out any technical errors in sequencing or influences from local sequence variants that might not be representative of the species, I analyzed TLR15 sequences from more than 100 wild Eudyptes penguins from all over their natural ranges. So the colors on this map indicate the species from which these samples were derived in the various different populations. And what I found was that TLR15 has been pseudogenized at least 8 times independently in the Eudyptes genus. And I also noticed that some mutations were widespread across the genus. They were shared by multiple species, whereas others appear to be more species restricted. And this may give us an indication about when each of the pseudogene haplotypes arose. So the pseudogene haplotypes that persisted across species boundaries may be much older than the more species-restricted haplotypes. But the most unusual thing that came from analyzing these sequences was that I also found intact haplotypes. And by that, I mean haplotypes with no evidence of pseudogenization. So overall, there were around 25% of haplotypes that were intact. And this corresponds to around 10% of individuals with genotypes that were fully TLR15 intact, so homozygous for intact TLR15. And it was the intact TLR15 haplotypes that really interested and confused me, because there seems to be a bit of a paradox here. So on one hand, the presence of multiple pseudogenized haplotypes in a single genus implies that there's been a strong selection pressure, or at least a pervasive selection that's persisted for a long time across multiple species. However, the fact that we also have intact haplotypes and none of the pseudogenized haplotypes has reached fixation suggests the opposite, that the selection has been too weak to drive any of the pseudogenized haplotypes to fixation. So how do we reconcile these two observations? The key to understanding this was to functionally characterize the intact TLR15 to see whether it was indeed functional. So to do this, I cloned the intact haplotype from the Eudyptes TLR15, as well as the emperor penguin TLR15, the northern fulmar TLR15, and the chicken TLR15. And I then expressed these as flag-tagged constructs in an NF-kappa B reporter cell line and stimulated them with either yeast lysate, which is a known agonist of TLR15, or another agonist that I tried for reasons that I'll explain later, which is aspergillus lysate. And as you can see, the emperor penguin, the fulmar, and the chicken respond very well to both of these agonists, whereas the Eudyptes TLR15 didn't signal at all. So this implies that there's one or more mutations in the intact Eudyptes TLR15 that prevents it from functioning. So to investigate this a bit further, I also created chimeric TLR15 constructs, the first of which is a constitutively active TLR, which works by fusing the extracellular domain of mouse CD4 onto the tear domain of TLR15. So the CD4 domains should dimerize, which brings the tear domains into close proximity, and that causes them to signal even in the absence of an agonist. So as you can see, the emperor penguin and the fulmar both signaled well in this chimera, but the Eudyptes chimera didn't function at all. So this implies there's a lesion somewhere in the tear domain that prevents it from signaling. The second chimera I made was a fusion of the Eudyptes extracellular domain onto the chicken tear domain, because we know that the chicken tear domain functions well. Again, though, the chimera didn't signal, which implies there's a further lesion somewhere in the extracellular domain of the Eudyptes TLR15 that prevents it from functioning. And altogether, these results suggest that there's at least 2 function-affecting mutations in the modern intact Eudyptes TLR15. So to summarize what I think's been going on, before the Eudyptes TLR15 was functional and responded to fungal agonists as it does in other species. And then in the ancestor to the Eudyptes genus, TLR15 acquired at least 1 loss of function mutation, but importantly, it's a mutation that didn't cause a truncated gene. I've just put it in the extracellular domain for the sake of argument, but it could have been anywhere in the gene. So TLR15 at this point is a cryptic pseudogene. Oops, sorry. And then because the gene is suddenly removed from purifying selection pressure, it acquires all sorts of other truncating mutations. In other words, the gene is overtly pseudogenized several times. And since none of the circulating TLR15s are functional, there's no advantage of any one haplotype over another. So the original cryptic pseudogene, perhaps with another function-affecting mutation as indicated by the work with the chimeras, still circulates to this day in wild populations. And this explains why we see a gene that has apparently been pseudogenized several times independently. In reality, it was probably pseudogenized once, but only as a cryptic pseudogene, and has only secondarily been disrupted and truncated. So what does the loss of TLR15 mean in practical terms for penguins? Well, in my functional work, you may remember I talked about aspergillus lysate, and aspergillus is a pathogen of significant concern in penguins. It causes aspergillosis, which is obviously a fungal disease that affects the lungs. So this is courtesy of one of my co-authors, who's a veterinary pathologist, Simon Spiro. It causes thickening of the air sacs and widespread granulomas throughout all of the viscera, and it usually leads to death. And you can see from the histology the extent to which infectious spores are produced by conidiophores in the air sacs, meaning infectious particles can be exhaled from an infected individual. if TLR15 is involved in Aspergillus immune responses, then it's likely that TLR15 could provide a genetic basis for aspergillosis susceptibility. So I need to do more functional work to see whether TLR15 is functional in other penguin genera. The nature of a cryptic pseudogene means it isn't necessarily apparent just from looking at the sequence whether it is or not. In addition, I need to do some epidemiological work to see whether penguins with functional TLR15 fare better than penguins with a TLR15 pseudogene in Aspergillus infections, or the other way around indeed, as Adrian talked about. TLRs are a double-edged sword. So I'll leave it there. I'd just like to thank my team of co-authors, in particular Michael Vinkler, who helped us out a great deal with several parts of this paper. Simon Spiro, veterinary pathologist, who provided me with the Aspergillus samples, and also Juliana Viana, who provided me with lots of TLR15 sequences from various penguins around the world. So thank you very much for listening, and I'll happily take any questions.